train, because what exactly happens is, okay, after finishing 1, 2, 3, and 4 clinical trials, so what happens is, okay, the virus will definitely must not recombine with the vaccine serotype, vaccine epitope. Okay, so the whole world is looking on that. After the vaccine is out, okay, the vaccine, because all the vaccines prepared, most of the vaccines prepared for this COVID-2 is based on live attenuated vaccine for spike protein. Okay, people are praying that by the time the vaccine comes, this spike protein should not mutate or recombine and change its structure. Okay, so if that happens, again, the whole vaccine trial should go back to ground zero. Okay, so if you look into the genome, it's just 29 to 30 kilobase pairs. Okay, this is a comparison of SARS-CoV-2 and MERS-CoV-2. There are only 2 open reading frames. There are several open reading frames, but only 2 open reading frames code for the structural and non-structural genes. So what exactly is happening is there are only 29 codable genes in out of 2 open reading frames. Okay, so out of 29, 27 are functional and 2 are non-functional. Okay, so we have got 30,000 genes in a human's body. Okay, so these 27 genes, okay, is equal to, it's like just like your Pandavas and Kauravas. Okay, 27 genes versus your 30,000 genes. Okay, these 27 genes has got a capability to interact with all the 30,000 genes in the human beings. I'm pretty sure this is very funny. This kind of interaction, protein-protein interaction, does not occur in any viruses reported so far. Okay, so this is a report which I read from a Nature paper, Nature Medicine. Okay, I found it very peculiar that how does a protein, how does 27 proteins interact with all the? Wait a second, my battery is gone. Wait a second, my battery is gone. Okay, I'm back. Okay, so it's a $1,000 million question. Okay, so why are the other SARS-CoV-1 is not able to interact with so many proteins? Because I'm talking about the insertion that has taken place from pangolin to, from bats to the human SARS-CoV-2. That insertion has really made a lot of difference in terms of the genome insertion recombination. Okay, so if you look into this part of the viral factors, okay, S protein is 2 types, S1 and S2. Okay, the spike protein. The S1 determines the viral host range. Okay, and S2 mediates the viral host cell membrane fusion. So if either of them, if S1 and S2, either of them is mutated, okay, I'm pretty sure infection may not occur. You forget about ACE2. ACE2, that is the second level. If any of them, either S1 or S2, again mutates, okay, there is a great problem occurring at the next level, which we are talking about. Okay, so there are so many. It's because it's a positive stranded RNA. Okay, it's a protein responsible for the transmembrane transport of proteins. Okay, and look at here, all the bat, swine, civet, and mouse, all are binding to ACE2 receptor. Okay, in the human beings. Okay, so there's a major complication. I'll talk about the ACE2 receptor at a later stage. Okay, people, especially at 65 years of age and more than them. Okay, and people underlying any immunocompromised diseases like hypertension, COPD, chronic obstructive pulmonary disease, diabetes, cardiovascular disease, respiratory distress syndrome, septic shock, metabolic acidosis, coagulation dysfunction, multiple organ failure, septic shock. All these are going to pay an additional factors, host factors, due to the effect of SARS-CoV-2. Okay, so this is very interesting part. I think most of you must listen to this talk. Okay, so this is the cilia which I'm trying to show you. This is the cilia in our mouth near our epiglottis, wherever we get mucus. Okay, the cilia is there. Okay, but this cilia has really is very tough layer. Okay, mucosa, submucosa, and adventitia. Okay, this is the cilia which I'm talking about in this picture. This cilia. Okay. So once the virus gets inside, it has to cross through cervical lymph nodes, esophagus, trachea, bronchioles, and ultimately reach the bronchial alveolar macrophages in the lungs. Okay, so this is the region where it takes a lot of target region. Okay, so if you look into the sub-regions, mucus blanket, there's so much of mucus blanket, gel layer, sole layer. Okay, pseudociliated columnar epithelia, endothelia, lamina proposia, so many things, okay? And we are talking about the tissue here. Virus has to cross all these barriers and come to the tissue, okay? So if you closely watch this is the cilia which I showed here, okay? This cilia. This is the cilia, okay? This is the tissue which I talked about, and this is the endothelial membrane, basement membrane or endothelium or epithelium layer, okay? So the target region for viruses is your cilia, okay? This is how the virus gets acquisition from here, okay? And with the help of neuraminidase enzyme on the spike protein, okay? It goes and cleaves the sialic acid, orthosialic acid linkages on the cilia, okay? So it cleaves it very clearly, and this is the only coronavirus and H1N1 virus are the only viruses across the globe which has the capability to act as a molecular scissors and cleave these sialic acids linkages, okay? And no other bacteria and no other viruses so far has a mechanism to cleave this layer, okay? And apart from this viruses, there are so many bacterial pathobiomes waiting in the dockyard to pave an entry along with the virus. The virus opens it, and along with the virus, it gets an entry inside. And okay, and the bacteria will pass through these lineages. Okay, bacteria cannot cleave the sialic acid and has to slide down through these passages. Okay, and go through fibronectin binding region here. Okay, because bacteria can cleave the fibronectin binding region. Okay, from there only it gets translocated into the tissue and then the tissue invasion. Okay, then after the tissue, there's something called bloodstream. If it enters the bloodstream, the virus becomes viremia and bacteria becomes septicemia or bacteremia. Okay, so there are so many viral dissemination and so many molecules are getting infected and so many altered mucus. Okay, so when there is infection of virus, we get mucus, repeated mucus, and we try to expel the both the virus as well as bacteria, but it's not possible. Okay, so that is reason. Okay, so this is the structure which I showed in my initial slide, and this is the mucus cilia. Okay, and this virus is marked as a pink spot on the mucus layer attached to them. Okay, on the epiglottis layer. Okay, and if you look into this slide, especially particularly, okay, so as far as my knowledge goes, okay, so all the carbohydrates so far, plant, animal, bacterial, yeast, mammalian, all the avian, all the carbohydrates so far existing on the globe are produced in the cytoplasm via glycolysis. Okay, but here, this is a long chain glycoprotein polysaccharide. Okay, it's a glycoprotein, it's a transmembrane domain glycoprotein. This is the only glycoprotein produced inside the nucleus. Okay, with the help of several silyl transferase enzymes. Okay, and post translational modifications and polyadenylations. Okay, all these things occurs, and while going into the nucleus, this is nothing but 5-acetyl neuramic acids, okay, or sialic acid or orthosialic acids, okay. These sialic acids become a secretive molecules. Even the cytoplasm does not know what's going on inside, what's happening, because all these monomers will form a chain, and then finally all these polyadenylations and all these things takes place inside the nucleus, and coming out of the nucleus, okay, it goes and reports to, look at here, it goes and reports, this is how it goes to it's a extracellular space, okay. It's a Golgi, the endoplasmic reticulum, and the cytoplasm is intracellular, is a nucleus. These nucleic acids, these O-linked and N-linked sialoglycans, they're called as O-linked sialoglycans or gangliosides. And we are talking about all the mucus layer is made out of orthosialoglycans. These glycans are transported into the nucleus, and inside the nucleus, all these modifications are occurring. And from here, with the help of silyl transferases, they get transferred from endoplasmic reticulum to cis Golgi and trans Golgi. And from trans Golgi, it moves from intracellular space to extracellular space in the form of endosomes or clathrin-mediated endocytosis or clathrin-mediated endocytic vesicles. Okay, if you look into the Barged Entry, okay, so there are so many commensal versus pathogenic bacteria. Okay, so along with the viruses, so many bacteria waiting outside along with the viruses to get pave an entry through mouth, pharynx, respiratory region, and then through skin, and then through urogenital tract, and through stomach, and the target organ for pneumonia and streptococcus aureus or Pseudomonas aeruginosa, cystic fibrosis, all these bacteria are very closely related to viruses. SARS-CoV-2, the target organ is lungs, and all of them will start mounting the pathogens and virulence at a time with a single knockout. Okay, so what happens is um, all these organisms together. Okay, virus, I'm pretty sure all are thinking that virus is the actual culprit to kill this people. Okay, so I'm pretty sure virus within the incubation time of 1 to 14 days or 1 to 21 days, it will kill as many as red blood cells and get out of the cell. Okay, and there will not be any imbalance. There will be some kind of imbalance between fresh red blood cell production and old red blood cell destruction. Okay, so that balance will be restrained, revoked only if you have a very strong immune system. So here what happens is the virus will live in a healthy immune system after 14 days, and bacteria will start uh, picking up switching on the virulence and pathogenesis, and then with the help of quorum sensing mechanism, okay, it will start playing, taking the control of the whole immune system, and by then it kills the whole system. I'm pretty sure it's not the virus, it is the bacteria coming along with the virus is killing the. They are called as secondary infections or nosocomial infections or hospital-acquired infections. These infections are acquired particularly uh, in the respiratory ventilators. Okay, because I don't think any ventilators in India has got a capacity or system to sterilize them, because they are very costly, nearly at 10 to 15 lakhs. So they cannot be sterilized, and they can be only blown with an air or washed with some small fluids, but not with they cannot be completely sterilized. And all the infections, secondary infections, are put with ventilators, and they're definitely going to stay there for some time. And then if it is put to the next patient, definitely they all are going to get killed with help of secondary infections. Okay, so these are the infections which are getting along with the viruses. They evade the immune system. This is the place which I showed you. This is the cilia, okay, mucus, and this is the bacteria, streptococci, and this is the Staphylococcus. This is the again some other bacteria. Okay, gram-negative Pseudomonas aeruginosa. For both of the target is the lungs. Okay, so after there is a breakage of sialic acids through viruses, the bacteria will pave an entry. Okay, and initially they colonize as a pioneers. Okay, they initially they start adhering to with the help of molecular bridges, and then they become settlers. Short-range adherence and high affinity specificity is produced. Society, they become a society, okay, with help of environmental signaling and exopolysaccharide formation, emulsifiers, biosurfactants, and lipids and glycolipids, okay, quorum sensing, and finally they become a community. The society becoming a community. Once they become a community, okay, they are going to take the whole of the whole cell signaling system, co-aggregation, metabolic synergy, and a lot of genetic exchanges takes place with the help of horizontal gene transfers and lateral gene transfers. So this early stage of colonization, and many of these factors will definitely promote the tissue invasion and blood invasion, and become septicemia and bacteremia or viremia. Okay, so look at this slide which I was showing about previously. This slide B. I showed you the slide A here. I hope you remember. We showed about the tissue here, and now the virus has crossed all these layers, and now it is in the tissue part. Okay, so this tissue now it is entered virus infected tissue. So now there's a tissue. Underlying to the tissue, it's nothing but your endothelium, epithelium, and your blood layer. Okay, so virus infected tissue has so many dendritic cells, interferon alpha, interferon gamma, cytokines, chemokines, neutrophil recruitment, deploying so many decoy molecules from the immune system. Okay, so both are together. Bacteria is also there, virus is also there. So bacterial proliferation and infiltration, just like your military people deputing them, and so many apoptosis will be occurring in the tissue, and pyroptosis, apoptosis, or whatnot. We have seen so many things here. Okay, so macrophages, dendritic cells, so many things are occurring, and once they alter the TLR, toll-like receptor pathways, and so many things are occurring here, and then finally they enter the bloodstream. If it enters the bloodstream, that means the person is no more. The person will not survive anymore. So if at all, if any survivors are there so far till date, have not crossed the tissue to the blood. If it crosses the tissue to the blood barrier, okay, that's over. Doctors have to pack the body in the plastic case, and send them outside. That's all. Nothing can be done. Okay, how does this COVID-2 hijacks your cells and kidnaps the signal transduction mechanisms? They're covered with spike proteins and they're entering into a vulnerable cellular environment with the help of an ACE2 and TMPRSS2. I'll talk about that. And releases a viral RNA, then hijacking the cellular machinery and protein machinery, okay, and making the viral proteins with the help of a cellular nucleus, and then assembling the new copies, all of them assembling in one particular place, spreading the infection. And finally, while exiting the cell, it erupts as a virus after virus after virus after virus. So like that, within few seconds, few million healthy cells will be infected with the help of the virions released from the infected cell. Okay, so if you look at the genome, I was talking about SARS-CoV-2 genome encodes 40 viral proteins. Look at here. There are 40 viral proteins only on the spike protein, which means the binding to AC2, okay? And the molecular weight of it, okay? So if you look into the similarity with the SARS-CoV-1, okay? With COVID-2, 87% similarity. In terms of E protein, envelope protein and matrix protein, membrane protein, it's 96%. But in terms of spike protein, it is only 87% similarity. Okay, why is SARS-CoV-2 more infectious? I told you it's a multi-basic cleavage site on the spike protein of SARS-CoV-2 is essential for infection of human lung cells. Why only lung cells? Because it's the tissue tropism for the lungs is very much specific and it will infect only lungs. So this pandemic virus harbors a highly cleavable S1, S2 cleavage site not found in closely related coronavirus. So this kind of S1, S2 polymorphism is not found in any other coronavirus, even in SARS-CoV-1. Okay, this cleavage at this site is mediated by furin, this furin receptor binding, and it's required for viral entry into the human lung cells. Okay, this is one major thing is nothing but a furin receptor. The S1 and S2 is going and binding on the furin receptor. Okay, so here what happens is your SARS-CoV-2, your lung infection, your intestinal has also got a lot of mucus, your lungs has also got a lot of mucus, your lungs have got two kinds of pneumocytes, alveolar type 2 and type 1 cells, okay? And type 1 cell is involved in exchange of gaseous materials like oxygen and CO2, and type 2 cells are involved in basically blood circulation and, okay, and blood pressure regulation, and there are so many properties, okay, especially permeability, increases the permeability of the alveolar cells, and okay, so vascular dilation also occurs with ACE2, okay, breathing, inhaling, very good breathing, very good inhalation occurs with ACE2. If ACE2 is not there, okay, so definitely we are going to face so many complications. And finally, your cytokines and chemokines are released immediately. Okay, your dendritic cells will come into the picture. Your ACE2 is completely depleted in even intestinal, gastrointestinal region. And immune cell activation, there's so many T effector cells, B cells, T regulatory cells. We'll try to stop it. We'll try to make some resolution. We'll try to suppress the hyperinflammation, okay? And if it is failed here, okay, actually there's so many mediators like cytokines, chemokines, tumor necrosis factor, IL-17, IL-2, 6, 7, 8, and growth and granular colony stimulating factors, GCS, of all these bodies are going to mediate between both of them, either to stop, either to go, either to proceed. Okay, so either to resolve the issue, because there's this big thunderstorm occurring in the lungs, and lungs are absolutely not visible. It's completely cloudy. Okay, they're trying to arrange some talks between both of the Treg cells and other cells, cytokines. Okay, and then here phase 1, phase 2, and phase 3. Look at here, the disease severity is completely causing them to cause cytokine storm syndrome, acute respiratory distress syndrome, monocytopenia, lymphopenia, ARDS, acute respiratory distress syndrome, shock, kidney injury, organ failure, secondary infections. So all these are going to happen. This is called pathogenesis. Okay, I'll talk about that at a later stage as well. So they encode several proteins to block the interferons. Interferons are biological antiviral agents. Okay, they are not chemical. So even they try to block them. So what I mean to say is, SARS-CoV-2 has completely understood the signaling pathway in the system. It pretty well knows about the immune regulations, and all these proteins are going to block the interferons. And the open reading frames actively blocks the induction of type 1 interferons. However, in SARS-2, the OR3b is truncated due to premature stop codon and hence susceptible to type 1 interferon. So SARS is not going to care any interferons, alpha, beta, and gamma. Okay, there's no worries at all. Okay, so if you look into here, okay, if you look into the exposed and unexposed conditions, okay, so there is a balance between CD4 and CD8 cells. See, if you look into the general flu symptoms, general flu, okay, unexposed, okay, even in unexposed, CD4 100%, CD8 is 70%. But in exposed, CD4 is 50% and CD8 is 20%. That means if the exposed people with the SARS-CoV-2, it is depleting the CD4 and CD8 cells like anything. So this kind of situation occurs only in the AIDS patients and immunosuppressive patients. Okay, so even if you give them anti-G, IgG and IgA titers, okay, the vaccine efforts are not going to, because I'm pretty sure vaccine or any drug system should be given to people, okay, whose CD4 and CD8 cells are like this. If there is a CD4 cells of 50% and 20%, I'm pretty sure even the vaccine is not going to work, because there's a lot of cross-reactive T cell recognitions going on between CD4 and CD8, and this is completely going to be undetected. Okay, so in any of the tests as well. Okay, something called recently in America, you can see that multisystem inflammatory syndrome. Okay, people initially said that only children will not be affected, and only elderly people of 40 and 65 age will be infected. So children, no problem. Okay, but children have recently suffered with some multiple inflammatory syndrome of the gut. Okay, so their pain, vomiting, diarrhea, neck pain, rashes, bloodshot eyes, or feeling extra tired. These association has been observed with SARS-CoV-2 infection or close contact with COVID-19 patients. Okay, so children are basically infected with multi-system inflammatory syndrome. Okay, only in children, nobody else, and that in only in US. So I talked about this stage 1, 2, and 3. So this is how the type 2 alveolar cells are largely infected, and they get disrupted, and apoptosis occurs outside the cells. Okay, I'll show you about that. So this is a Nature paper come in 2020. Okay, so this is a TMPRS receptor. This is a ACE2 receptor. A TMPRS receptor is nothing but a serine protease receptor. Okay, here ACE2 receptor and TMPRS2. Okay, in either of them, one helps in binding, one helps in activation. If either of them is ACE2 or TMPRS serine protease receptor is missing, infection does not occur, and vascularization or endocytosis may not occur. Okay, and finally, okay, people may be survived. In the worst case, okay, leakage of vascular permeability and dysfunctional immune response. Okay, so all these interleukins, chemokines, monocytes will be, because once the virus gets inside, okay, they all secrete for a positive feedback. Okay, for a positive feedback, it goes to a cytokine storm, and okay, and from here, if you want a healthy immune system, I told you your CD4 and CD8 cells, okay, should be very active, and it should be more than 60-70%. If it is 50-20%, okay, this is going to be like a dysfunctional immune system. Immune response is going to occur, and cytokine storm is going to take place, and hyperinflammation and hypoxia, and cells will be deployed of oxygen, and completely pulmonary edema and pneumonia will be a secondary stage, and where bacteria will take over from virus and start running inside the lungs. So there is an immunological imbalance in COVID-19. I told you CD4, CD8, and okay. Report in Lancet says ARDS is a common immunopathological death event that's occurring. It's reported in Lancet 2020. Okay, I'm telling like cytokine storm, the deadly uncontrolled systemic inflammatory response resulting from the release of large amounts of pro-inflammatory cytokines, interferon alpha, gamma, IL, beta, 6, 12, 18, 33, chemokines. They're all mediators. They try to fix a deal between good and bad, and they really try to do the best. Okay, growth stimulating factors. And this cytokine storm can trigger a violent attack on the immune system, the body, causing the ARDS and multiple organ failure, and finally leading to death in the severe case of COVID-2. And people are saying this, only 80% of people will not lead to the death, because they will not cross the blood barrier. Only 20% of people whose mucous layers is completely depleted due to various reasons of diabetes, hypertension, or smoking, or alcoholic, or cystic fibrosis, bypass surgeries, and all these people will be fall under 20%. Okay, so if you look into how the COVID-19 is affecting early stages, look at here, the virus paves an entry into the lungs, immune cells, the presence of virus activates the immune cells to fight the infection. Look at here, the two kinds of cells, type 1 alveolar and type 2 cells. I told you the surfactant layer is lining the alveolus inside the lungs. With the other type 1 cells helps in exchange of gases of oxygen and CO2. Okay, so this layer is largely infected and affected, and there's a lot of damage causing on the this layer, and surfactant is completely, because we need surfactant because this is air sacs. Okay, we need surfactants to play a greasy role, to inhale and exhale. While inhaling and exhaling, it should be lubricated. Okay, if this lubrication is not there, okay, this lining gets injured, and obviously CO2 or oxygen may not take place. Okay, and obviously this will pave an entry, inviting so many mediating cells like chemokines, cytokines, and these air cells will be infiltrated with so much of plasma inside, and this plasma will invite so much of trouble, invites some pneumonia, streptococcus pneumonia, because pneumonia needs like a swimming pool inside. Okay, so pneumonia will be, because it's filled with so many mediators. Okay, so cytokine storm, just like the thunderstorm occurs inside. So this is the protein rich fluid. I told you it's a biosurfactant produced inside. Loss of surfactant, and the fluid is filled with interstitium, and neutrophils are deputed inside, and all the cells are deputed, and becomes a protein and cellular debris and formation of a scary tissue. So there is no tissue, this becomes a scary tissue. Okay, so this is how a healthy lungs alveoli should be. And once it takes up the virus, it becomes C. The surfactant looks like an yellow fluid. Okay, all these surfactants will be depleted. Okay, vasodilations increase the permeability of so many infiltration occurs. Okay, so immune response will be completely down, and that is how once it gets inside you through ACE2 receptor. Okay, and direct activation of lectin pathway, complexes of classical pathway, toll-like receptors, Treg cells, RIG1 pathway, MD5A pathway, C3 complement fixation pathways or alternate pathways. And all these will invoke the responses from neutrophils, macrophages, and finally it is called as a cytokine release syndrome, and accumulation of neutrophils and monocytes and macrophages. Look at here, day 0, your ACE2 receptors are inhibited, and on 1 or 2 days, the replication will start working very well, pretty well in the airways, in the pneumonia, okay, in the alveolar cells. And from day 3 onwards, your innate immune system in the lungs and maladaptive inflammatory response. Okay, this is where, and finally, after 14 days, it gets into ARDS, acute respiratory distress syndrome. And if it crosses here, that means it has gone into the bloodstream. Okay, so we have got mucus in all the layers of gastrointestinal tract. You have got your mucus in the skin, oral cavity, respiratory tract, your urogenital tract, your vaginal tract. Okay, so your virus is prone to every place wherever there is mucus, and I'm pretty sure even the virus was traced out even in the stool samples recently. Okay, so very invasive infections, all the invasive infections spread at the target other organisms like blood circulatory systems with the help of septicemia or viremia. Okay, they even cross the blood-brain barrier, even they cross the olfactory portal and brain barrier portal. And once you're infected with the CO2, COVID-2 infections, okay, at least for 3 months, a person will have a liability to lose a sense of smell and a sense of taste, because we have got nearly around 600 G protein coupled receptors on our body and our cells to sense at least so many tastes and so many masalas, so many non-vegetarian foods. Okay, all that capabilities will be, this olfactory bulb, the receptor nerve cells linking to them will be completely null and non-functional. Okay, so I was talking about the COVID infecting other organs. Okay, even it can infect the blood clots, even it can cause the blood clots, blood vessels will be clotted. Okay, thrombotic events may occur. Okay, and they cause life-threatening complications, especially pulmonary embolism, heart attacks, strokes. Okay, all these can also occur due to COVID-2 in a severe complicated patients. Okay, so let me just cross these slides. And, yeah. So, yeah, there's something called immune system overdrive. I told you, once the virus gets inside, the T cells, the B cells, and nearly CD4, CD8 are depleted. Oxygen is depleted, hyperinflammation, hypoxia occurs, and so many neutrophils, monocytes, macrophages are recruited, and T cells, and so many things. A cytokine storm is occurring. Okay. So with this terminology, we may get to learn so many new terminologies in immunology or immune system overdrive. Sometimes even we come to know if you want to travel abroad, we must be having a passport, okay? And sometimes from now on, they'll ask you immune passport. Do you have an immune passport against this? Okay, I don't have immune passport, so immune passports has to be made. So after COVID-19, okay, there are several new immune terminologies will come into picture, okay? So the cells will undergo protective responses or reactive responses, okay? So hyaline membrane of the proteins and dead cells of alveoli making the breathing difficult or impossible. These cytokine molecules will stimulate the movement of cells towards sites of inflammation, infection and trauma. So alveoli air sacs with oxygen transfer from lungs and the bloodstream will be mostly affected. Okay, so this is a picture of a lungs. Okay, there's so many nonsenses occurring inside the lungs with the help of deploying of decoy molecules, of chemokines, macrophages, dendritic cells, delayed expression of interferon gamma, because it knows pretty well about the interferon gamma is not going to affect on the virus anymore, and it's going to affect the TLR, toll-like receptor, CCR receptors, blockers, and all these signaling pathways, MODs, ARDS, T cells, apoptosis. All see, virus is well known about every step of these pathways. So it is very much affecting the pulmonary and epithelial endothelial cells. Okay, induction of high levels of cytokines. All these things are occurring inside only in the lungs, and lungs will be like just like a mashed potato after the infection. Okay, so all these virulence factors once enters inside the cell, they alter the host cell signaling, and okay, so they do a lot of apoptosis, proliferation, disruption of epithelium integrity, and so many things are occurring. So much of hijacking of signaling transduction takes place with the help of this bacteria along with the viruses. Okay, so these are so many events that are taking place inside the cell. Okay, early endosomes and late endosomes and this and that. I think there is a lot of biology to talk about. I'm pretty sure I'm given less time. I'm the last speaker, and I think I'm not be able to talk completely on this topic today. Okay, so this ACE2 receptor, attachment receptor, okay, for CoV1 and CoV2, both the receptor is same, and TMPRSS2, it's nothing but a serine protease, cellular protease. This spike protein priming the cellular protease. The neutralization efficiency, look at here. The neutralization efficiency is very high in CoV1, but very moderate in CoV2. So this is the reason why we are losing so many cases. Okay, yes, if you look at here, ACE2 is the receptor for activation. Okay, this is attachment, and TMPRS2, serine protein, is this attachment is activation. If one of them is mutated, virus cannot come and bind here. Okay, so both should be there and both should get an entry. Okay, so both should help in taking, because it's ACE2. Otherwise, in a human system, I told you, without ACE2 we cannot breathe. Without ACE2 there is no blood circulatory system. Without ACE2, because all the blood pressure tablets which you're taking, it goes and binds on the ACE2 receptor. Okay, so what this virus does, while going and putting on itself on the receptor, it while taking it endocytosis with the help of while going inside, it takes the ACE2 receptor inside along with them, and mostly what happens is that the number of ACE2 receptors on the cellular surface will be completely depleted.
Dr. Veera Brahmachari?
Yes,
would you be able to wrap up in about 5 to 6 minutes?
Yeah, I'll do that. Yeah, even I'm trying to do that. I'm in that
place. Okay, thank you. I'm looking at that. Yes, thank you.
Thank you. Yeah. So let me just finish this slide. It's very complicated. Okay, so this is alveolar epithelial cell, and so here what happens is, so SARS-CoV-2 is getting inside. It's this tumor necrosis factors, all the factors on the cell. Okay, colony stimulating factor, interferon gamma, and interferons. Okay, all these pathways. Okay, with the help of TLR7, it recognizes them. Okay, and JAK pathway and STAT3 pathway are going to majorly infect them. Okay, and finally, okay, it's going to hamper all these signaling transduction pathways, inflammasome complex, capsase complex, and all these complex infected, okay, are completely loosed. And then if any of these factors are, any of these pathways, especially JAK and STAT3 and NF-kappa beta, okay, if NF-kappa beta, STAT3 and JK are the major signaling transduction pathways, if these three pathways, these three pathways will lead to inflammatory genes. If these three pathways are overproduced, okay, hyperinflammation may occur and cytokine storm may get into picture. Okay, so that is how your infection gets into the blood receptor. Okay, so I think if I go much beyond this, because it's a hardcore pathophysiology, hardcore biochemistry, and hardcore molecular biology. So I'm pretty sure most of the students out there are just graduate students. I don't know if they have really understood because it's. Let me wind up here, madam, because I'm pretty sure I have much more slides to talk, but I'll end up here, madam. Yeah, I'm free to take up any questions quickly at this minute, madam. Yeah, I'll stop my talk here.
Yes, I know,
Yeah.
but very interesting. I do understand. Even for a layperson like me, I've learned so much more than any other, YouTube videos that I've seen, Dr. Veera Brahmachari. I must say a big round of applause to your patience. At the end of the day, after being tired, right? But I think Dr. Manishri will ask those questions. But let me quickly ask before she asks. Dr. Rekha Rani Kokanti is asking Dr. Veera Brahmachari, there are many mutant strains related to COVID now. What is the criteria in developing a vaccine which will be used all over the globe?
I'm really shocked. See, without knowing the most circulating virulence phenotype or strain, how do you go ahead preparing a vaccine? I know the target is spike protein. I know the target is AS2 protein in the human cells. Okay, but I'm pretty sure the vaccines has to be prepared completely on the most circulating strains, but not on any strain picked up from that epidemic or endemic region.
Yes, you're right.
Yeah, because if at all, if you prepare a vaccine, people are saying vaccine will come this Monday, that Tuesday, this Saturday. I'm pretty sure vaccine, if it comes, vaccine comes only and works only on that particular community, but not an Indian population. Because our population is different, their receptor is our receptors are different, you know.
Yeah, very true, correct.
Mutations, spike proteins, there's a lot of, I'm pretty sure it has to be, all the strains have to be sequenced, there should be thorough study, then only the vaccine has to be prepared.
You're very, very correct. In fact, she's again asking, so what's your comment on reinfection in recovered COVID patients?
See, you must be having 30 to 40% neutralizing antibodies in your body after infection, okay? But I'm pretty sure you must not be reinfected with a new serotype again. If you reinfect with a new serotype again, you are under trouble.
Yes.
Get it?
True. And she's the same one asking, how far immune target therapy is effective in treating COVID?
Immune therapy, again, I think the patient who is suffered and recovered should be able to give his consent to give a good amount of plasma or convalescent plasma for the treatment. I don't think anybody, they will be already weak and I don't think they'll be giving blood. Because I'm pretty sure who gives blood is a very healthy individual recovering must be able to give blood. But already person who has come from 21 days of bed, quarantine bed and ventilator bed, I'm pretty sure he must not be even having enough energy even to talk.
Yes, I know, correct. You said very, very well. My question, it's my own personal question, Dr. Veera Brahmachari. I'm so impressed with your presentation. I hear a lot of people saying that, COVID, this virus behaves differently with different people in different countries. And, it is different in India. And what is your take? You've explained so beautifully.
Yes, comparatively, see, when I talk about myself, I'm a very, very healthy individual. And I'm pretty sure I'm confident. I'm confident on my immune system. Even if infected, I can come out of it because the kind of genes what our forefathers have given to me and the kind of immune system I am carrying on myself. Okay, so that varies from person to person. Because supposedly, if at that time your immune system is pretty good, and you are suffering from fever and down with running nose and kind of thing, if you pick up the virus at that juncture, okay, the chances of pulling your immune system much more down. See, that means at that time, if you're infected with a common cold already, and your CD4, CD8 cells are down below 50%, and then if you're infected, if you're picked up with a new virus, That
True.
means even though you're a healthy individual, you're going to see the bed for 21 days.
Yes. No, that I agree. But my point was, when the virus enters the body, it mutates. You know that mutation is going on. It mutates differently for different people. Is that something which people are saying?
Yeah, that's what I'm telling. If your CD8 and CD4 cells are more than CD4, CD8 cells are more than 70,
Yes.
80, 100%. Okay?
Okay.
Virus is going to do its job 7 days and get away from there.
Oh my God. So I wish I had more of those. Yeah.
What I tell you is your immune system should be always good, and
Okay.
all the time you should be wearing masks and washing clothes and gargling, and all these things should be done frequently.
True. But I must tell you, before I hand you over to Dr. Manishri to propose the formal vote of thanks, on my own behalf, I must really congratulate you, Dr. Veerabrahmachari from Krishna University. You're amazing. I'm telling you very honestly, I've seen many YouTube videos being a social scientist, but I've not seen anyone so wonderfully clear, in-depth, and which is easily understandable by a layperson, probably like me, right?
So I'm also unhappy you did not let me speak for the whole presentation.
I wanted you to speak. No, we gave you 1.5 hours. We gave you your time. So you should not have a grouse on that.
On this good evening, I'll write in my diary, so-and-so college did not allow me to speak.
No, we gave you 1.5 hours. I've kept my time. See, just we gave you your quota of 1.5 hours. I made sure they had another link. I was there sitting, I said, no, I'll not allow Veerabrahmachari to go back with this. The young chap that he is, he has to talk for his 1.5 hours. But I tell you,
all three of us today who spoke are classmates, so we know each other, and we all are the same intensity.
That's so you shouldn't have a grouse if they enhance their time. Okay?
So they killed all my time.
Very good. No, really nice. Please stay. Just hold on till Dr. Manishri proposes the vote of thanks. Okay. Thank you so much. Okay. May I now call on Dr. Manishri to propose the formal vote of thanks?
Thank you, ma'am.
Please unmute your video, Manishri, so that we can see you. Thank you.
Yes, ma'am.
It's, I tell you, Veerabrahmachari, I've been doing just like you. From morning, I've been at a webinar, even till today. From morning 9, I've been here. So I've been working very hard.
Is there a part of NAC, madam?
No. Our college only propagates knowledge. We're not bothered about NAC. That's secondary.
That's great.
Manishri.
Thank you, ma'am. Thank you, ma'am. Sir, first of all, I want to thank you. I want to say thank you because you have given a detailed presentation about coronavirus. So once again, I'm telling you, thank you, sir. Thank you for your presentation. As now it's time for propose a vote of thanks. We, Dr. A. Veerabraham and I from the Department of Biotechnology express our sincere gratitude to the management of St. Joseph's College for Women for creating the environment and infrastructure required for this webinar. A special thanks to respected Sister Principal, Dr. Sister Saiji, for her unconditional support in organizing this webinar. Thank you, Sister. Our deepest gratitude we owe to our 3 eminent resource speakers, Dr. Gumma Vidyanidhi, Dr. Jogeshwar Gadi, and Dr. P. Veerabrahmachari, for spending their valuable time and sharing their knowledge with all of us and patiently answering all the questions. We thank you very much, sirs. We thank our Vice Principal, Sister Hema, who assisted us professionally in designing the webinar. We express our gratitude to respected Sister Casey Mary, Secretary of the College, for her support. We thank Dr. P. Sharada, IQAC Coordinator, for the academic circulation of this webinar on both academic and scientific community. We are indebted to Ramani Madam in arranging the required protocols for smooth conduction of this webinar and helping us at every stage of this webinar. Really a very big thank you, Ramani Ma'am. We thank other department faculty for their valuable guidance and suggestions in smooth conduct of this webinar. We express our sincere gratitude to the technical advisors of Mr. Nagasrinivas, Assistant Professor, Department of Computer Science, Dr. V. Gauri Shankar, Assistant Professor, Department of Mathematics, and Mrs. Swarnalata and Ms. Chandini and Ms. Lalita of the administrative staff for their technical support in designing and publishing the brochure.
Dr. Veera Brahmachari, just hold on, because I'll ask you 2 questions after she finishes, because
Sure.
Dr. Bindya is very keen on that.
Yes,
Manya, please continue.
We would be failing in our responsibility if we did not mention or if we did not thank all of you, our participants, without whom this webinar would not be a success. Thank you, everyone. Over to Ramni ma'am.
Dr. Veera Brahmachari, are you there?
Yes, madam, I'm there, very much there.
Dr. Bindya is asking,
Yes,
why people are forgetting TC and NK hybrids for targeted treatment of COVID?
Madam, TCN? What is
TC and yes, NK hybrids.
I don't know, what are they, madam?
I don't know, she has written, why are people forgetting TC and NK hybrids for targeted treatment of COVID?
See, I know
And she's
what
also
is TC.
asking, do you think the drugs being released so fast in the market help us in any way?
Drugs at the most will help you block the endosome complex, not going until nucleus.
Okay,
So not beyond anything.
not beyond
So
that.
hydroxychloroquine and recently dexamethasone has come up.
Okay. Right.
Feminapir, remdesivir, all these molecules are just only helps stop replication for a while, not to the depth.
Yes.
Because virus is much more intelligent than all of us and all the drugs
True.
and all the scientists living in it.
Very true, very true. So thank you, that will be all. And we really thank you once again, Dr. Veera Brahmachari. Sometimes after all this COVID, everything finishes, I will definitely see you somewhere, probably in Krishna University. I would like to personally meet you because you seem so young and dynamic and very impressive.
I'm 43 years old.
Don't worry, I'm 17 plus, so I'm 60. I just retired. Okay, so don't worry. I'll be a big competitor for you, okay, at my age. I must say you're really amazing. And all the 30 participants, we need to give them a big round of applause. They listened to every word till they came back and they still listen. That is what I would say, even if 3 people listen to you till the end, that's a victory won for you. And that's the best thing. That's the best
I have the confidence that I'll go on with my presentation. I'll keep
I was thinking, this fellow is really determined that he will talk for that 1.5 hours. I said, no. Afterwards, he may even call me and say, Ramni ma'am, what is this? Why didn't you were the big spoiled sport, like, when we cancel a match and all that. So I said, no, I will allow it.
I'm very stubborn. I'm very determined. Even when you speak, when you see me speaking directly,
yes,
the whole hall will be silenced, and I will make them keep silent.
Yes, Dr. Vibram was saying that. We are very, very fortunate. The world has more teachers like you, and wish you all the best. And may God bless you also with loads of energy, and to do a lot of research, get more awards. We want to see next time your profile with 10 more awards probably. And it's lovely to see you.
Thank you.
Right? So let's stand by for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय
Jai Hind from St. Joseph's College. Stay at home, stay safe. And we are very happy once again. We've concluded the biotechnology webinar organized by the Department of Biotechnology. Dr. Veerabrahmam and Dr. Manishri who worked very hard. And of course, our technical team, you see Dr. Gauri Shankar from the Department of Maths. And we're very happy with all the speakers today from this whole afternoon. All three of them, Dr. Vidyanidhi, Dr. Yogeshwar, and Dr. Our last speaker was so good. We are very, very happy that we had such good speakers. Jai Hind. And once again, join us in another webinar. It's bye for now. Good night. Take care. Stay home. Stay safe. Thank you.